eradication. Till here you have any doubt? Yeah? No? If you have doubt, then comment down. We will answer your comments. We will answer your queries. Now, make the heading. Another ways to achieve this sterilized, you can say, products or environment is to eradication. Probably you know that all the invasive procedure in the medical field is prone to get in the microorganisms. Suppose take the example of the endoscope. Okay, you are putting the endoscope inside the you can say a patient or a diseased person. So there is a chances that the bacteria will you can say colonize on those endoscope, isn't it? Or if you can say putting your stethoscope on the patient chest, okay, or patient abdomen, there can be chances to colonize the organism on this stethoscope, isn't it? And you know that you cannot provide the heat to the stethoscope, you cannot provide heat to the endoscopes. These are the heat, you can say, susceptible medical devices. So you have to look for certain another techniques where you are not providing the heat, you are providing something else, and you are achieving the sterile products, isn't it? So for that, there are techniques called as the eradication. Okay, the another technique is called as the eradication. So make the heading eradication. It's good. Now, for this eradication, what things comes in your mind? What do you think about this eradication? Yeah, we are not allowed to use the heat now. Okay, we are not allowed to use the heat now. Then what things we will use? Yeah, very good. We are now using the radiation. Why? What we are using? The radiation. I know that radiation is not harmful, you can say not useful, or it's not beneficial for the human. It's harmful. I know, but these eradication methods, they are meant for only for the sterile environment, as well as this is practiced under the you can say the why wearing the proper equipments. By wearing the proper equipments, this eradication procedure is applied. Fine. So we are using the two different types of radiation. One is called as the one is called as the ionizing radiation, and another one is called as the non-ionizing radiation. The first one is ionizing radiation. Okay. Another is called as the non-ionizing radiation. Now, this non-ionizing radiation, it uses the UV rays. You know that natural sunlight is one of the most available sterilization technique for all. It's for God grace, isn't it? It's the nature grace. So already by the help of the UV rays which is present in the sun, okay, they make bacteria under their non-pathogenic state. Okay, their non-pathogenic state. So the same UV radiations we are using, and what we are doing, this wavelength of this UV light is, you can say, 260 nanometer. How much? 260 nanometer. So we are heating the object, sorry, not a heating, we are radiating the objects, okay, by using the UV radiation. And this UV radiation, you know that all the living organisms, they have their genetic material, which is, genetic material is universal, isn't it? Genetic material is a universal. It is present in the everyone. Okay, every living creature. I'm talking about the living creatures. So these UV rays, they are basically destroying the DNA. They are destroying basically their genetic material. Okay, you know that our DNA is made up of by the two nitrogen bases. Okay, called as the purines and pyridines. Called as the purines and pyridamines. So by providing the UV radiations, okay, these UV radiations, they goes to the they goes to this DNA. You know that this DNA have the nitrogenous bases like adenine, guanine, cytosine. In the case of RNA, it's a uracil. Okay, so these nitrogenous bases, okay, these nitrogenous bases, or you can say the purines and pyridamines, they form the dimers. Okay, they form the dimers, and by providing this UV radiations, their DNA is damaged. Their DNA is damaged. So this is the one way to destroy the microorganism, their normal cellular function. And you know that the genes are present over the DNA, and genes are those which program the cellular function, which direct the cells to, you can say, function optimally, okay, to function optimally. So if you damage their DNA, a bacterial, or you can say a viral DNA, it means you are completely disturbing or impairing the bacterial function. Thereby, bacteria will not survive and they will destroy. Now, but there is certain drawback. There is a certain drawback associated with this UV radiation. Not all bacteria will completely destroy by this UV radiation. Now the question arises, why? Now, in the presence of light, bacteria undergo photoreactivation. Photoreactivation. Bacteria have certain enzymes. Okay, bacteria have the certain enzymes which bacteria use and again make their genetic material, you can say, functional. Again, bacteria make their genetic material functional. Fine. Some of the bacteria, not most all the bacteria. And then why we are using this? We are using just because viruses are highly susceptible for this UV radiation. If you destroy by the UV radiation to the viruses will destroy. So viruses are susceptible. Viruses are susceptible to the UV rays, or you can say UV light. Viruses are susceptible. Fine, no problem, easy to understand. And it is, remember, it is harmful for the human. So we have to practice these techniques under the guidance, or you can say under you can say wearing the personal protective kits. Fine, no problem. Now, the another technique is the ionizing radiation. What things we are doing? These ionizing radiations, they have the high wavelength. We are using the high wavelength rays. Okay, the one of the commonly used rays is called as the gamma rays. Okay, called as the gamma rays. Now, this gamma rays property is that they can penetrate deeper. They can penetrate deeper. Okay, they can penetrate the deeper. So basically, we are using these gamma techniques, gamma rays, in the hospital setting to sterilize the catheters. Fine. To sterilize the syringes. Okay. To sterilize the rubber globes. Okay. What things? Catheter, syringes. Okay. Rubber globes. Fine. So this is called as the non-ionizing radiation. Now, comes to the another technique. The another technique is called as the filtration. You understood this sterilization by means of eradication? Yeah? No problem? Okay, once again I will recap it. Eradication means to using the certain rays, okay? Using the certain rays, or you can say using the radiations, we are completely destroying the microorganism. Fine? There are two techniques behind this eradication. The one is called as the ionizing radiation. Another one is called as the non-ionizing radiation. In the non-ionizing radiation, we are using the UV rays and their wavelength is 260 nanometer. Fine. These UV rays, they goes to the bacterial or viral genetic material and they disturb their, you can say, normal sequence of their genetic purines and pyridamines, okay, their nitrogenous bases. Thereby, their cellular function is, you can say, impaired, and bacteria or virus become non-functional. Got the point? But one of the, you can say, drawback, or you can say one of the cleverness of the bacteria is that bacteria have certain enzymes which, under this light, they undergo the photo reactivation, and by which they repair their genetic material. But viruses are susceptible to this UV rays. They will destroy themselves. While we are bombarding to the UV rays to the virus infected, you can say, the materials, viruses will destroy. The non-ionizing radiation, in the non-ionizing radiations, we are using the high wavelength rays. We are using the high wavelength rays. Okay, and one of the rays is called as the gamma rays. One of the rays is called as the gamma rays. And these gamma rays, the property is that they penetrate deeper inside the material. And for that, we are using the catheter, we are using the syringes, and we are using for the rubber globes. But remember, this eradication is must practiced under the, you can say, protecting a practitioner, because these are the harmful. They are called as the carcinogenic. They can cause the cancers in the human. So they are the lethal. They are the death causing. Now let's move and discuss the last, you can say, techniques for achieving the sterilization is called as the filtration. Commonly, these filtrations we are using in our day-to-day life, isn't it? We are using. The last techniques we are discussing is filtration. Filtration. You know, if the materials are not withstanding the heat, if we are, you can say, you worried about the harmful or lethal effect of this radiation, then what can we can do, you can say, alternate to achieve the sterility. The one is the filtration, to make, you can say, so minute pores that bacteria or you can say viruses will filtrate, they not comes into the products. That's the one way. Okay, we are not now providing the heat, we are not radiating the, you can say, the product, we are just filtrating. Means we have to make certain minute pores, okay, in the filtration slit, by which bacteria will not come. Okay, bacteria will not allowed to come. They are you can say they are filtrated. Okay, so for this filtration, they are mainly applied for the liquid, such as the solution of antibiotics or certain components of culture media. Okay, so basically we are using these filtrations for liquid media, okay, liquefied antibiotics. Okay, and why? How? The filtration pores. Filtration pores are very, very minute. Okay, very, very minute. No problem. Now, so I will first draw the diagram how you are that filtrating the liquids. See, this is the membrane. This is a membrane, and it have very minute, you can say, pores. Very minute pores. Okay.
Oh, we would create out in the concrete,
it have very minute pores. and this is the vessel. Now, and here there is a, you can say, provision for clearance of vacuum. So it is here for the vacuum. Now, this is the membrane, and these membrane, this is continuously asked in the competitive exam, so you should remember these membrane, they are basically made up of by nitrocellulose, and it can be made by the polycarbonate. So nitrocellulose and polycarbonate, okay, these are, you can say, certain material which we are used to prepare the membrane, to prepare basically the filtration membrane. And now here you are pouring the liquids, liquids comes, okay, it's come inside through the vacuum, the gases are, you can say, become outside, it goes outside, and here you achieve the sterilized liquid. Got the points? So this is the one way to achieve the, you can say, sterilized product. Fine, no problem. Now, generally we are using the chemical for disinfection, isn't it? We are using the chemicals for the disinfection, and sterilization is basically the physical process. It's basically the physical process we see by the means of heat, by the means of rays, by the means of filtration. But there are certain, you can say, gases which act as a sterilization. If you employ those gases, you achieve the total microbial encounter. Okay, you can achieve the total free, you can say, product from the microorganism. And one of the notable is the ethylene oxide and formaldehyde. So apart from the heat, eradication, filtrations, there are chemical ways. To achieve the sterilization. The chemical sterilization, the one you just keep in your mind, okay, is formaldehyde. This is in this we are also using to preserve the specimen, isn't it? To preserve even the autopsy body, okay, in the autopsy center, as well as the ethylene oxide. Ethylene oxide, formaldehyde and ethylene oxide. These are two gaseous. Yeah, these are two gaseous, you can say, chemical ways to achieve the sterilization. Fine, no problem. And there are liquid sterilization as well. You just keep in your mind liquid sterilization. These are the gaseous. If we talked about the liquid, then it's called as the glutaldehyde. Fine. Glutaldehyde. Apart from the glutaldehyde, something called hypochlorite. Basically, these are used in the linen cleaners. You know, the hospital, all those, you can say, like curtain, bed sheet, you can say the gowns, all are called as the linen. Okay, the hospital clothes are basically called as the linen. So for cleaning of those linen, this glutaldehyde and hypochlorites are used, and these are basically the chemicals. Or you can say basically these are the gaseous, these are the liquids. So these, if in exam, if they will ask that about the glutaldehyde, you just remember, and hypochlorite, these are the liquid chemicals sterilization techniques. And if you did ask about the formaldehyde or ethylene oxide, they are the gaseous chemical sterilization. So after having said all these things, let's summarize today's class. what we studied in the today's class, we basically studied about to how to control the microorganism, and in that we discussed about the goal, the aim, why we are controlling the microorganism. Okay, then we discussed about the various methods, okay, and two methods which we have to complete as far as our syllabus is concerned. One is called as the sterilization, another one is called as the disinfection. So in the today's class we have discussed about the sterilization, different method of sterilizations, like the heat, okay, like the eradication by the means of radiation, by the filtration. We discussed about some of the chemical sterilization method. Now in the next class we will discuss about the last part of this lecture is called as the disinfection. So thank you with having me. Bye. Have a nice day.